Hello everyone, welcome back to my video, and in this video I am going to solve the KSET lessons question paper from 2013 to 221. What I did is I have collected all the questions which are asked in the KSET from 2013 to 2021. I have made a list here of the topic cellular organization, that you can say cell membrane. Okay, so what you have to do is, after studying the topic cell membrane, you just have to go through these questions so that you will get to know from which area is more important for the examination, from where they have asked the questions, so that will enhance your marks. Okay, so that is the reason. Okay, I have made this. Now let's start with the first one. Okay, so the first question is that which of the following is not true. They are asking which of the following is not true for facilitated diffusion. Okay, so they are asking about the facilitated diffusion, which is not true. So before answering this question, let's have some knowledge about this facilitated diffusion. So we know that in diffusion there are two types, that is a simple diffusion and then facilitated diffusion. So first, in simple diffusion, in which particles are moving from region of their higher concentration to a region of lower concentration. Okay, that is also same for the facilitated diffusion, but there is one difference in that this facilitated diffusion is going to occur, or the process is going to occur by means of helper proteins. That is the main point here. This process is going to occur by means of helper proteins, like it is something like a carrier mediated transport. Okay, and it's then if you have to say more difference is that simple diffusion is occurring in prokaryotes and facilitated diffusion is going to occur in eukaryotes. Okay, so as I said before, this simple diffusion occurs directly through the phospholipid bilayer of the cell membrane, and this facilitated diffusion occurs through helper transmembrane integral proteins called carrier proteins or channel proteins. Okay, and if you have to mention some of the particles, those which can cross this layer, is that in case of simple diffusion, small and non-polar molecules like water, oxygen, carbon dioxide, ethanol, urea, these can transfer in case of simple diffusion. Coming to facilitated diffusion, like large polar molecules. Okay, remember the large polar molecules like large biomolecules like glucose, ammonia acid, nucleic acid and charged ions can transfer the membrane in case of facilitated diffusion. Okay, and there is no inhibitor molecule can inhibit the process of simple diffusion, but for facilitated diffusion can be inhibited by a specific inhibitor. Okay, if we have to give the example for simple diffusion, is that diffusion of respiratory gases such as oxygen, carbon dioxide, this is the example. And if you have to give some more example, like the animals which can get rid of their waste materials or waste products like urea and uric acid, carbon dioxide, this is the example for the simple diffusion. And example for facilitated diffusion is that there is transport of glucose like amino acids, nucleic acid inside the cell. Okay, this is the example for the facilitated diffusion. And then one more example if you have to give is transport of sodium, potassium in and out of the cell. That is the example for the facilitated diffusion. Okay, this is the some basic knowledge about the facilitated diffusion. Okay, and let us look at the options here. A requires cellular energy for the transport of substances. B requires a special carrier molecules to move substances across the membrane. C allows only non-polar molecules, not the ions. D option shows hyperbolic kinetics. See, let us analyze these four options here. First one, requires cellular energy for the transport of substances. They are saying option is A like that, and that give a more focus to the question here. They are asking not, which is not true for the facilitated diffusion, right? So so if you before that, if you have to say, there are two types of transport, right? One is active transport, another one is the passive transport. Okay, so in case of active transport, energy is required in the form of ATP, that is cellular energy. Okay, for the active transport. But for the passive transport, there is no energy is required. There is no requirement of cellular energy, that is ATP. Okay, and this active transport is against the concentration gradient, means molecules are moving from low to high. But in case of passive transport, it is along the gradient, means higher concentration to a lower concentration. Okay, and if you have to give the example for active transport, sodium potassium pump and proton pump are the example for the active transport, and diffusion and osmosis are example for passive transport. Okay, as this simple diffusion and facilitated diffusion both are passive transport, so as I said, now passive transport doesn't require any energy, so it doesn't require cellular energy for the transport of substances. But here they are saying requires a cellular energy for the transport of substances. That's why this is not true. Okay, option A is the correct answer. And coming to option B, requires a special carriers for molecules to move substance across the membrane. Yes, it requires a special carriers, like as I mentioned before. special carriers like carrier proteins, channel proteins are required to cross the membrane in case of facilitated diffusion. Okay, that's why this is not, but this is correct for the facilitated diffusion, but they are asking which is not true. Okay, that's why this is not correct. Okay, then C is coming to C option, allows only non-polar molecules, not the ions. So as I mentioned while giving the difference between simple diffusion and facilitated diffusion allows the larger polar molecules, okay, not the non-polar molecules. Okay, that's why this option is also correct. They are, because they are asking not true. Okay, and shows hyperbolic kinetics. For hyperbolic kinetics, you should know the one experiment, theory, that is Michaelis-Menten kinetics. Okay, there you are going to see this hyperbolic kinetics. Okay, so they are saying here it shows hyperbolic kinetics. Actually, they are asking as it is. They are asking not true. Okay, here they're saying shows hyperbolic kinetics, which is not correct for this facilitated diffusion, because facilitated diffusion doesn't show this hyperbolic kinetics, but they're saying it shows, and they are asking not true. That's why for this option, this is not correct. Okay, I hope you understood. Don't get confused, because they are asking here not, which is not true. Okay, if you didn't understood, just go through these points once again. Okay, now which is asked in the year, the KSET in 2013. Okay, now if you understood that first one, let us move on to the next one. Next one is second one. The second question is like this: membrane rafts are examples of, which is asked in the year 2015. Here membrane rafts have another name, that is it is also called as membrane rafts. It has another name, that is also called as lipid rafts. Okay, these are also called as lipid rafts, both are same. Okay, so as we say, this lipid bilayer, see for make you to understand, see I have drawn the diagram here. So sorry, I forgot to show this. Now I am showing this simple diffusion. If you show the simple diffusion, this is the cell membrane. Okay, and this is outside the cell, and this is inside the cell, and this cell membrane is composed of many kinds of molecules like sphingolipids, glycosphingolipids, and they are rich in phospholipids and gangliosides and carbohydrates, proteins. Okay, fats, lipids, everything will be present in the cell membrane. So it is not homogeneous. Okay, it is heterogeneous. Okay, and so for simple diffusion, see the diffusion particles are crossing the cell membrane. Okay, for example, if you have to take the water like this, if you have to differentiate, you can look at the facilitated diffusion. In case of facilitated diffusion, they have the channel proteins and carrier proteins to transfer these diffusing particles from outside the cell to the inside the cell. Okay, this is the difference. So now as we started with the second one, let's continue the second one. So these membrane rafts are also called as lipid rafts. Okay, and here they are asking example for the membrane rafts. Before that, let us know what are these lipid rafts, or membrane rafts are this this bilayer. Okay, this bilayer is not homogeneous, and it shows localized variations, like some variations, it shows some localized variations. Okay, or you can say it shows some localized variations, and those are called as microdomains. Okay, and those are called as microdomains, and these microdomains again divided into ion induced aggregations. Those are called as lipid rafts and caveolae. Okay, these are nothing but variations, localized variations. Okay, some variations, so what we call them as microdomains. Okay, and these divided like this. So they are asking examples here. So if you take the first option A, option A is membrane vesicles. We know that the vesicles, these are called as vesicles. So these are not the examples of for membrane rafts. Okay, this is not the correct. And the second option is membrane microdomain. Just now I explained. So this is the correct answer. And polarized membrane, no, this is not the answer. And Catherine coated vesicles. These Catherine coated vesicles are these are comes in the receptor mediated endocytosis. Okay, there are types of endocytosis. In that this is receptor mediated endocytosis. Here you can see or you can say this as a Catherine coated pit mediated endocytosis, you can say. So these are pit like invaginations. Okay, invaginations of the plasma membrane that are coated with a network of protein called as Catherine. That's why these are called as Catherine coated pit mediated endocytosis. There I will that comes under the endocytosis. So this is not also example for membrane rafts. So the example membrane rafts are examples of membrane microdomains. Okay, what are membrane microdomains? These are nothing but localized variations. Okay, now let us move on to the next one. The compound that binds metal ions and capable of ion transport, which is asked in the year 2015. Okay, options are like this. Option A, ion core. Option B, isoprenes. Option C, interference. Option D, introns. Okay, before solving this question, let us know the meaning of these four options. Okay, because some of the questions doesn't require an in-depth knowledge, but if you have a little bit of knowledge also, you can solve these problems. Like for option D, you can take the option D, that is introns. So now start to remember where you heard this word introns, whether this introns is related to this topic or not. Okay, introns we have heard while studying the transcription and translation, right? This intron is nothing but this is a non-coding section of an RNA transcript. Okay, it is a portion of a gene that doesn't code for any amino acid, that is introns. That's why this is not the correct answer. Okay, coming to next C option, interference. Interference, where you are heard, maybe you might be heard in the immune system, right? In the immune system, they are heard where these interference are going to attack the which causes attacks the viruses. Okay, these interference are some of will be man-made. Okay, so this is not the correct answer. Next, option B, isoprenes. So isoprenes, this is as industrial. These isoprenes are industrial production of synthetic applications. These are going to produce for in industries for the production of synthetic applications like rubber for fires, tires. Sorry, and these isoprenes are colorless volatile liquid. Okay, it's like a rubber. So that's why this is also not a correct answer. Okay, and the left one is ionophore. And you may observe here the transport of ions. Okay, in the name, ion, pore means a hole. Okay, so the compound that binds the metal ions and capable of ion transport is ionophore. Okay, that is the correct answer, and it is asked in the year 2015 of the KSET. Now let us move on to the fourth one. The next question is that the two important functions of the cell membrane are. So they are asking two important functions of the cell membrane. For this, you should know the function of the cell membrane. So which is asked in the year 2015. Here, the option A is selective permeability and active transport. Option B is selective permeability and partial transport. Option C is active transport and permeability. Option D is passive transport and permeability. So we know that if you have to solve this, if we know that active transport, passive transport, okay, these are all the functions of the cell membrane, and permeability is also. But we also know that cell membrane doesn't simply sense all the molecules. Okay, it is going to select. That's why. So these combinations are not correct. Okay, and if you already select selective permeability means the transmembrane is going to allow only selected molecules, right? Okay, and they're saying here the partial transport. No, the partial transport is not going to take place. That's why the correct answer is it is selective permeability and active transport. Okay, and these two are the important functions of the cell membrane. Now let us move on to the next question. The next question is that fifth one. Question is like this. The question which is the exclusive component of mitochondrial membrane. Options are, which is asked in the year 2016. Options are option A cholesterol, option B phosphatidylcholine, option C sphingomyelin, option D cardiolipin. So they are asking exclusive component of mitochondrial membrane. So for that you should know the topic here. There is no logic here. You should go to the components of the mitochondrial membrane. So for that for this answer the correct answer is option D. This is the component, that is cardiolipin. Okay, which is asked in the year 2016. Now let us move on to the next one. The next one is question is like this. In the beta cells of the pancreas, proinsulin is converted to insulin. Okay, which is asked in the year 2016. Option A, where it is converted, they are asking that where this proinsulin is converted into insulin. Okay, in the beta cells of pancreas. So in the beta cells of pancreas, in that location, this proinsulin is converted into insulin. Okay, so where this process is happened? So option A, in the bloodstream, option B, in the catherine coated vesicles, option C, in the Golgi complex, option D, at the time of fusion of mature granula. The correct answer for this is in option B, in the clathrin coated vesicles. So these clathrin coated vesicles, as I was explaining the previous question, there I said these clathrin coated vesicles are these are nothing but receptor mediated endocytosis. Okay, so if you take the endocytosis, okay, what is endocytosis? Endocytosis is the process by which cells takes in the substances from outside of the cell by engulfing them in a vesicle. Okay, in that there are three types, that is phagocytosis, pinocytosis, and receptor mediated endocytosis. So for the third one, the receptor mediated endocytosis, for that example is the catherine coated pit mediated endocytosis. In that nothing but these are pit like invaginations. If you have to say like this, see to explain this, these are like this pit like invaginations. Okay, like this pit like invaginations of this plasma membrane that are coated, and these are coated with a network of protein. Okay, coated with a network of protein called as clathrin. Okay, so there only this proinsulin is converted into insulin. Okay, that is the correct answer. Now let us move on to the next question, the seventh one. The seventh one, and the question is like this. The mechanism in which the rate of solute movement increases by interaction of transmembrane proteins is termed as option A, endocytosis, option B, simple diffusion, option C, facilitated diffusion, option D, active transport. And this question is asked in the year 2016. So I will repeat the question and try to understand the question here. The mechanism in which the rate of the solute, the question in which the rate of the solute movement increases by interaction of transmembrane proteins is termed as. See, as I explained the facilitated diffusion, see, you can look at in the diagram facilitated diffusion. In facilitated diffusion, it is different from the simple diffusion. Okay, simple diffusion occurs just crossing the, so it doesn't require any channel proteins or the carrier proteins. But facilitated proteins require the channel proteins and the carrier proteins. These are these channel proteins and carrier proteins are nothing but the transmembrane proteins. Okay, because it it will help to helps the diffusion particles to to cross the membrane. Okay, these are the transmembrane proteins. Okay, so the question what they are saying, the mechanism in which the rate of solute movement, and this solute movement is going to increase by interaction of transmembrane proteins. So by interaction of transmembrane protein, what is this process is called as I explained before, endocytosis is a totally different function. So because endocytosis is the process by which the cells are going to intake the substances from outside of the cell by engulfing them in a vesicle. Okay, that is endocytosis. And simple diffusion, just now I explained. So active transport, it is active transport is a process. Okay, this is also not correct. So option C is the correct answer, that is facilitated diffusion. Okay, that is the actually this is the definition of the facilitated diffusion. Okay, now let us move on to the next one. So the next one is again they are asked on the membrane microdomains. Okay, the eighth one, lipid rafts and caveolae are examples of these are the examples of what? So as I explained before only, these are the examples of membrane microdomains. Okay, and which is asked in the year 2013. Okay, so very so you can say this question is repeated but indirectly. That's why I said after you studying completing the subject, after studying the subject, go through these previous questions. Okay, so that you will get to know how the questions have been formed and how questions have asked and which are the important questions, so that questions that are repeated, so that that will help you to enhance your mark. Okay, now let us move on to the ninth one here. Where are the ion carriers located in a cell? As I mentioned before, ion carriers obviously they will be located in the cell membrane. Okay, they will be located in the cell membrane. For example, you can see now if you say these are the ion carriers, okay, these ion carriers will be present in the cell membrane. Okay, in the cell membrane, the ion carrier will be present. Okay, these are all wrong. Intracellular space no, nucleus wrong, cell wall is wrong. Okay, cell in the cell membrane. Now let us move on to the next question here. Which of the following are the essential requirements for Michaelis chemiosmotic hypothesis to work? Okay, the option A here, the option A is the inner mitochondrial membrane should be intact. Okay, which of the following are essential? They are asking the essential requirements for this Michaelis chemiosmotic hypothesis to work. If this has to work, what are those requirements they are asking? And they are given the requirements, and we have to choose the correct requirement here. Okay, so here the correct requirement. The first they are saying the inner mitochondrial membrane should be intact. Means what they are saying, if you have to know the requirement means for your key information, what I would like to mention is you have to know the requirements means you should study this experiment, that is Michaelis chemiosmotic hypothesis. You should have to study, then only you will get to know the requirements. Here the first requirement they are saying the inner mitochondrial membrane should be intact. Here yes, this inner mitochondrial membrane is, we know that this is the, if it is a mitochondria. Okay, the mitochondria, this is the outer membrane, this is the inner membrane space, and this is the inner membrane, right? So this inner mitochondrial membrane should be intact means it should be in a original condition, it shouldn't be teared or damaged. Okay, so to study this, the inner mitochondrial membrane should be intact. That is a correct option. And second one, there should be a proton gradient between inside and outside the mitochondrial membrane. Yes, if you have if the ions or molecule should have to cross the membrane means there should be a proton gradient, right? Okay, then the next option, the inner mitochondrial membrane can be permeable to these ions, they are saying. So this is not correct option. Okay, so they might be permeable, might not be permeable, but it's like that is not a necessary, like they should be permeable. Okay, and the next option is pH outside the inner mitochondrial membrane will be same as that on the inside. So this is also not correct. Otherwise it's not like it should be same. Okay, so if you have to find the answer for this, so what could be the correct answer? Which is the correct option? If you look at here, the C option contains the first and the second. Okay, that is the correct answer. Now let us move on to the last question, which is asked in the year 2020 of the KSET, and a question is like this: which of the following pair matches with the membrane transport process and its primary function? Okay, it is asked in the year 2020. Okay, the option A, exocytosis, the movement of macromolecules into the cell. Okay, So here exocytosis, they are saying the movement of macromolecules into the cell. They are saying into the cell. Exo means outside. Okay. This is not the correct one. Next one. Pinocytosis. Pinocytosis, what is this pinocytosis? Pinocytosis means the uptake of water and solids into the cell. It means uptake of water means like uptake of fluid, you can say. Fluid also you can say. Pinocytosis, but this is not the primary function. Okay. And coming to osmosis, passive diffusion of small solids. Okay. And phagocytosis, secretion of large particles from the cell. Okay. So, but the primary function of the see, these you can say pinocytosis and phagocytosis, these will comes in the endocytosis. Okay, even this is the it includes the membrane, but still these are not the primary functions, and exocytosis is also not the function. Okay. So the correct option is osmosis. Okay, osmosis, that is passive diffusion of small solute. Okay, this is the correct answer. So these are all the questions which I have collected from year 2013 to 2021. Okay, these are the questions which have been asked in KSET Life Science. Okay, I hope this video is helpful for you all, and if you learn something from this video, please do subscribe and share this video. Thank you everyone.